Facing the daunting challenges, the Micro, Nano, Total Analytical Systems Lab provides the link among engineering, biology, and chemistry.
Reactors at production scales are bulky. Using miniaturized unit operator helps us to rapidly develop a new manufacturing process due to its high heat mass transfer and short reaction time, especially at downstream process, which requires a lot of extraction and separation steps in multi-phase solvent system. In our lab, we developed several techniques that allows two immiscible liquids to be separated and extracted effectively, and without using complex setup or pressure control components.
For example, biodiesel is the product of trans-esterified methanol and soybean oil. Through the process, it will form to two immiscible liquid, biodiesel and byproducts. In order to accelerate and make safety for the two-phase liquids separation and extraction process, we developed a core annular flow device using stretchable helix wire placed at the core position. One classic example is the biodiesel manufacturing. Individual ingredients such as methanol, biodiesel, catalysts, and byproducts stay in two immiscible phases. Using our technique, less interfacial tension phase, usually organic phase, preferentially wets the wire and is subsequently propelled out of the wire by the incoming flow, decreasing the distance between the aqueous droplets and causing them to accumulate inside the helix wire. The accumulated aqueous droplets eventually form a continuous cylindrical stream and are radially encircled by and flow towards the outlet. Unreacted methanol can thus be separated from the mixture for sustainable use of resources. This novel liquid-liquid separator can completely separate immiscible mixture without any complex pressure regulator. It also adapts to both liquid-liquid and liquid-gas system. Similar liquid-liquid interface control method is applied on the liquid-liquid extractor. We manipulated the length and the gap of the helix wire to keep two-phase stream flowed core annularly and established a stable contact interface for high-efficiency mass transfer. Such controllable residence time and contact area help us to precisely estimate the system overall volumetric mass transfer coefficients for scale-up process.
The key advantages of these two unit operators compared with other methods are that it uses adjustable wire gap that offer large operating windows. In terms of flow rate and flow radius to adopt various extraction model systems and easy to fabricate and assemble.
These wire gaps are much larger than process of membrane-based method, and therefore no fouling or bulk edges occur. Our unit operator can thus be used as a standalone unit with automatic regulation and be coupled with other upstream and downstream unit operators.
In addition, we introduced machine learning method to predict the performance of the liquid-liquid separator, extractor, or other micro-unit operator. It conduces to save the time of process investigation and consumption of the materials.
There is growing interest in the biology and chemistry community to increase automation of laboratory and process systems to facilitate feedback control, optimization, automate screening, and data collection. The advantages of using lab-on-a-chip (LOC) microfluidic devices include cost-effective, smaller size, low volume of samples, less skilled manpower, reusability, and environmentally friendly. LOC techniques allow miniaturized and integrated devices to perform on-chip process automation to reduce the time and manpower. For example, phage screening process includes the mixing for affinity binding, filtration, and collect the target phage. Using integrated microfluidic reactor, valve, and filter device can finish the screening process automatically. We also collaborate with biologists and chemists to develop instruments for researches. This homemade electrode has a similar transendothelial electrical resistance measuring trans to the commercial electrode. In the transwell plate experiment, and good stability that is stable due to the continuous measurement in the incubator directly.
Microchannel has a higher mass and heat transfer and help to decrease the reaction time.
Integrating the actuator and the sensing technique to an automation system could reduce the manpower and achieve more precise process control.
Additionally, the NanoTotal Analytical Systems Lab also establishes visualization technologies from the physical characteristics of liquid flow and the subsequent engineering design. These techniques can further strengthen and expand new scientific questions. Sharks are apex predators. They smell prey that are hundreds of meters away. We proposed a hypothesis of stereosensing and somatotopic map of animals that have never been tested after examining the structure and form of the shark's head and nasal cavity. Odor's movement along the fluids passively in the cavity reflects external time and spatial information, which helps them locate and track distant prey. The life-size models of a blue shark's head and the nasal regions and computational fluid dynamics are achieved using reverse engineering scanning, microcomputerized tomography, and magnetic resonance imaging techniques. Red dye solution indicated the odor's flowing trajectory near shark's nasal structures and inside the nasal cavity. Particle image velocimetry technique also shows us the flow around the shark's head. This could help us to study effects caused by head structures of sharks and their swimming behavior, including pressure distribution, velocity field, and the incurrent and exterrent nostrils of nasal cavity. Professor Zhang's group also discussed the time and spatial characteristics of the fluids entering nasal cavity and how they are affected by morphology and behaviors. Numerical computations on a simplified 3D microfluidic model can answer these much complex questions and eliminate the defeat of morphology reconstruction. Odor trajectories and partial odor concentration map could be studied.
The function of nasal structure and the evolutionary strategy of shark for chemical sensing are known. A bio-inspired chemical sensing, detecting, and checking system is developing.
For different purposes and specifications, and optimal tuning can be found. Learning from nature, we could always find other solutions for encountered problems.
The NanoTotal Analytical Systems Lab is small lab but complete in every detail of fluidic field. We keep dedicating fluidic manipulation technique in different scale and solve problems through self-development. Microfluidic-based bioanalysis system for engineering, biology, and chemistry studies, and transfer deep science innovation into world-changing solution.